Okay, so make sure you do the homeworks fairly soon after doing these, because this is going to get forgotten pretty quickly. And I do not blame you for having difficulties memorizing the clotting factors. If I remember correctly, they're numbered based on their discovery, not based on how they come up. Okay? And so there's some really bullshit things that happen there. Anyways, please work on them. Let me know if you have any questions. There's other ways we can approach the material and talk about it, okay? So we can go through those questions and figure it out. Okay, so this is DNA, RNA, and the flow of genetic information. Of course, the dogs mixing and doing all sorts of weird genetic offspring, right? Okay. All right, so the central dogma of biology, essentially. If you ever wonder why the Russians did not have great hospitals and stuff, even though they really were amazing like engineers and space programs and stuff, they did not accept the central dogma. They accepted an alternative to the theory of evolution with genetic information. Okay? So the DNA and RNA, these are long linear polymers. These are called nucleic acids. The nucleic acids are going to carry information in a form that can be passed from one generation to the next. No, you do not have to have DNA to pass your information on. There are ones that have just RNA. Okay? We have DNA, of course. The central dogma is the flow of genetic information from DNA to RNA to proteins. That's the central dogma of biology, essentially. And you probably know this and have been told this, what, since 111 or earlier, right? Yeah. Okay. And that's why it's the central dogma, because you're doggone sick of it, right? Okay. So this right here is the DNA. It's going to be replicated. So we have the blueprint. And then what we're going to do is we're going to go from DNA to RNA, and this is going to be transcription. Now notice, because they are both very similar in structure and very similar in bases, right? We call this transcription, i.e. transcription as in you're like transcribing a book, right? It's still a book, it's just maybe in a slightly different formatting, right? Okay. And then RNA is going to go into protein. This is going to be translation, right? Translation, because this is a fully different language. We don't have the 4 bases anymore, do we? Okay? Okay. Now, a nucleic acid is going to consist of 4 different types of bases. These are going to be linked to a sugar phosphate backbone, your deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. The linear polymers that are well suited to function as carriers of genetic information, that's the definition for DNA and RNA that your book proposes. Okay? And it's a good one. A nucleotide is going to be each monomer unit within the polymer. Now, notice the language here. Nucleotide, that is a monomer of them, right? So one monomer, it is a set of 3 things in a nucleotide, right? So each nucleotide will be composed of a sugar, a phosphate, and one of 4 bases. Okay? And that's the monomer that composes the backbone. Nucleic acids, on the other hand, are going to be what you compose that entire monomer, or polymer, excuse me, out of. And that's going to be your strings of DNA and RNA, right? Okay, so the polymeric structure is right here, represented in an abstract. This is the sequence of bases. It's going to constitute a form of linear information that you can then read. And this is how you would represent this: base I plus 1, base I plus 2. And you're starting to get used to that, hopefully. Everybody clear on what that says and how to read it? Fantastic. Note that as you read down, you get the sugar, phosphate, sugar, phosphate, sugar, phosphate for your backbone with the bases turned in towards each other. So the sugar component is different depending on if you're looking at RNA or DNA, but only a little bit. So ribose is the sugar in RNA. That's why it's R in RNA. This has a 2' carbon atom of the sugar that's linked to the hydroxyl group. The deoxyribose is the sugar in DNA. And so the 2' carbon atom of the sugar is linked to a hydrogen atom instead. It does not have the OH group there. That absence of the 2' OH is going to increase the resistance to hydrolysis. Remember, alcohols were sites that we can do hydrolysis on, right? And so by removing the alcohol, we get a more stable backbone that's less likely to change, right? But note, on that same vein, this is ribose. Ribose is a what? Sugar. Yeah, it's a sugar that you'd find in people's bodies. Okay? This is not just ribose, is it? This is deoxyribose. This is a sugar that has actually had a reaction happen to it that would require some degree of energy and, expense. Okay? And so DNA is expensive, isn't it? So, by the way, this is the 2' here. Everybody see that red OH, red H here? Okay. And anybody know what the 5' and 3'? That's how we usually depict the directionality, right? Yeah. And this is the 3' by the way. This is the 5'. Okay. And this one right here would be the 1'. We'll get more into that when we talk about sugars and the polymers they make. This right here is a pointing upwards 1', or a beta. Okay. So again, the backbones of the DNA and RNA will consist of the sugars linked by phosphodiester bridges between them. The 3' of one sugar is going to be esterified to a phosphate group. This is then going to be joined to the 5' of another adjacent sugar, 3' here, phosphate alpha here. And then that'll be linked to the next 5', right? Which is this one right here. The backbone is going to be constant in a nucleic acid. Each phosphodiester bridge is going to have a negative charge. What this means is it actually repels the nucleophilic species that can hydrolyze a backbone. So normally you'd have a nucleophilic attack, right? Okay. But what are most nucleophiles? What charge would they be? Likes positive charge, it's probably negative, yeah. And so when we have these phosphate bridges, it's going to very much limit, just have a little corridor where you could have a negative charge possibly try to come and attack. Okay? And it's going to be repulsion all the way along, right? Okay. So it really does make this hard to attack, and that's why it's a good genetic information carrier, right? Because you've protected the outside of this damn thing. So here's the sugar phosphate backbones for our nucleic acids. This one's the DNA. If you ever don't have a label, how do you tell it? Yeah, you look for the base, 1', 2', hydrogen or oxygen. Okay. Okay, and this is RNA right here. Anybody have any questions so far? So the bases of DNA and RNA, the bases are going to be attached to the 1' carbon atom of the sugar. 2 of the DNA bases are going to be derivatives of purines, and these are the purine bases. Remember where we talked about purine versus pyrimidines? Okay, this is the purine. And so that's the adenine, A, and the guanine, which is G. 2 of the DNA bases are going to be derivatives of the pyrimidines. This is going to be C, which is cytosine, and T, which is thymine. Now the RNA does replace the thymine T with a uracil, U. And again, that is going to be a pyrimidine as well, okay? Again, it's pyrimidine derived. Now, this makes a lot of sense. One of these has 2 circles, right? Okay, 2 rings. The other has 1 ring, right? And so to fill up the same amount of space, what do we need to do? We need the same number of rings each time, don't we? And so you get a 2 to 1, 2 to 1. Okay? If you get a 2 to 2, you'd have 4, right? It'll take up too much space. If you get a 1 to 1 match, what would happen? Not close enough to hydrogen bond, right? Yeah. And that's what we're seeing. This is purine, by the way. This is pyrimidine. So the purine here is going to be the one with 2 rings in it. The adenine and guanine are the ones that are the purines. The pyrimidine, on the other hand, is going to be cytosine and uracil and thymine, of course. That are the pyrimidines. Anybody have any questions there? Okay. All right, so the nucleotides are going to be monomeric units of nucleic acids. Nucleoside is going to be the unit that's consisting of a base bonded to a sugar. The RNA nucleosides are going to be adenosine, guanosine, cytidine, and uridine. Notice again, there's a change in terminology, okay? We have the nucleoside. This is just the base bonded to the sugar. We don't have anything else. What's missing in that statement? Phosphate. Yeah, we don't have the phosphoryl, do we? Yeah. Okay. And so the DNA nucleosides will be slightly different. They'll have deoxy in front of them. So deoxyadenosine, deoxyguanosine, deoxycytidine, and of course thymidine, because uracil versus thymidine. We don't have to put a deoxy, right? Okay. If you were going to look at these, by the way, if you had A and you just wanted to represent it by an A really quick, like ATP, okay? What do you think DATP means? The deoxy form, right? Got it. And then DGTP. Yeah, and that one had the triphosphate, and that one has the energy that's ready to get linked, isn't it? Yeah. Okay, so a nucleotide, this is going to be a nucleoside, so we have all the stuff in a nucleoside, the base and the sugar, right? And then we have it joined to one or more phosphoryl groups. This is going to be via an ester linkage. The nucleoside triphosphates are going to be the precursors of the DNA and RNAs. Those are the GTP, ATP, all those, okay? Or DATP, DGTP, if we're talking about the DNA. Okay. Okay, so these are going to be N-beta glycosidic linkages that are linking these together, okay? And that's the base being linked to the sugar, right? So the C1 here, this is that sugar C1, is going to be attached to a nitrogen, which is the 9th nitrogen of the purine, or the 1st nitrogen of the pyrimidine. This linkage is called an N, because we have a nitrogen on one side, right? Then dash for a bond. Beta means the side. This is on the sugar, okay? And so it being above the ring of the sugar, the same direction as this part right here, is beta. The easy way to remember that is beta has a little tail, so it looks like it's higher. See that? If it was below where this hydrogen is, that would be alpha. Okay? And we'll talk about that more when we talk about like sugars linking together. Right now, it's kind of an extraneous thing that's kind of a side note. Okay? But know that we do the beta linkage, and they're always above the ring like that. Okay. Okay. Is that purine or pyrimidine? Purine. Yeah. Okay. Very good. Does anybody have any trouble remembering which is which? No? Okay. Fantastic. There is kind of a trick, but I don't think it works as well as just knowing and having it memorized with some flip cards. All right. So the DNA molecules are going to be very long. They'll also have directionality to them. They're always stacking upwards, right? And so your DNA molecules must be comprise many nucleotides to carry the necessary genetic information. Our E. coli genome is a single DNA molecule that consists of 2 strands, which are 4.6 million nucleotides each. This is actually DNA sort of spreading out, unwinding. Okay? So some DNA molecules can be extremely large. Are the humans the most complicated? in terms of genomes and DNA? Yeah, we're nowhere near the most DNA or genomes, right? Anybody know what has the most DNA and genomes that has sort of been found and studied so far? Due to our interference and things that are happening with us, corn. Corn has about 400 genomes in it. And in fact, species have been found with like 800 and doubles. Okay. We're going to go ahead and stop here because we are right on time. And we'll start here next time.